All right, let's jump right in. Today, we're taking a seriously epic journey. We're going to shrink ourselves down, way, way down, leaving behind the giant macro world we're used to, and we're entering the invisible, bustling, microscopic factories that literally power all living things on Earth. From the tallest redwood tree to the skin on your very own fingertips. The whole shebang starts right here. Have you ever actually stopped to wonder what builds a giant tree? Or a human being? Or even just a tiny little ant? It's kind of like a massive Lego castle. That giant castle is made of hundreds of tiny individual plastic bricks, right? Well, every living creature is made of microscopic building blocks too. But to really understand these blocks of life, we first have to figure out how to even see them. Section 1, the microscopic world. To get a grip on life, we've got to understand the tools that let us see it. 0.1 millimeters. So why does that specific number matter? Well, think about drawing two tiny dots on a piece of paper and slowly bringing them closer and closer together. At a certain point, your eyes just can't tell them apart anymore. They blur into one single dot. For the human eye, looking from a normal reading distance, that blur happens when things are about 0.1 millimeters apart. We call this the limit of resolution of the human eye. Anything smaller than that, we're literally blind to it without some backup. And that backup arrived in the form of the microscope. Way back in 1665, a scientist named Robert Hooke used this early light microscope he designed. It magnified things like 300 times. He looked at a super thin slice of cork and noticed these tiny box-like compartments. He actually called them cells because they reminded him of little empty rooms. Fast forward to today, and we've leaped incredibly far. We've got massive electron microscopes that use a beam of electrons instead of light. These absolute game changers let us zoom into the nanometer scale. That's 1/1000th of a meter, by the way, letting us see the crazy complex details inside these cells with crystal clarity. Section 2, crossing the cell membrane. All right, first stop on our journey into the cell, the outer wall. Every exclusive factory needs a bouncer, right? For the cell, that bouncer is the cell membrane or plasma membrane. The text calls it selectively permeable. Sounds fancy, I know, but if I'm explaining this to a kid, I just say it gets to play favorites. It decides exactly who comes in and who gets kicked out. It happily lets in the good stuff, like water and oxygen, while totally blocking the harmful stuff and keeping the really important machinery locked safely inside. Okay, let's actually test out how this bouncer works with a classic experiment from our explainer source text. Picture this. You take a kitchen knife and slice a potato into two identical pieces. It's super important they are the exact same size and weight from the jump, just so we have a perfect baseline. We drop piece number 1 into plain water. But piece number 2, we drop that into a beaker filled with a super concentrated 20% salt solution. I want you to really think about this for a second. What do you guess is going to happen to the water inside those potato cells after, say, an hour? All right, here's the breakdown. The plain water is a hypotonic solution, which just means it has fewer dissolved things in it than the inside of the potato cell itself. Because of that, water rushes into the cell, and our potato actually swells up and gets heavier. Now, an isotonic solution would mean things are perfectly equal inside and out, so zero change there. But that 20% salt solution, that's hypertonic. It has way more salt outside the cell than inside. Nature absolutely loves balance, so the water literally rushes out of the potato cells through that selectively permeable membrane to try and water down the salty environment outside. The result? The potato piece shrinks, loses weight, and gets all rubbery. It's osmosis in action, and it's pretty awesome. Section 3. Inside the cell factory. We made it past the bouncer. Welcome to the main floor. Before we wander around, though, let's figure out what kind of factory we're actually in. There are two main setups. First, prokaryotic cells. These are like your simple, tiny, one-room workshops. The pro just means primitive. Bacteria are prokaryotes. They're incredibly small and don't have a secure, walled-off office for their genetic material. But then, we have eukaryotic cells. These are the big leagues. These are the complex factories that make up you, me, and every plant outside your window. They're much larger, up to 100 micrometers, and feature a highly structured, well-defined boss's office that we call a nucleus. And here is that eukaryotic factory floor. See all those distinct, colorful shapes floating in that jelly-like fluid? That fluid is the cytoplasm, and the shapes are called organelles. That literally translates to little organs. Think about it. Just like your body relies on a heart, lungs, and a stomach to function, the cell has these specialized little structures that each do a very specific, absolutely vital job to keep the whole operation running smoothly.
Let's meet the crew. First up, the big boss, the nucleus. This office holds your DNA, which is the master instruction manual for literally everything the cell does. Next, the mitochondria. These guys are the powerhouses. They take the food you eat and convert it into chemical energy, ATP, keeping all the machines humming. And finally, we've got the lysosomes. Think of them as the cleanup crew, or like a microscopic recycling center. They're little sacks packed with enzymes that chop up and destroy worn-out parts or toxic waste. Fun fact from the text, they even help human sperm cells break through the outer layer of an egg during fertilization. Every single organelle has a job, and they all work together beautifully.
Section 4, Plant versus Animal Cells. Now, you and a leafy oak tree both run on complex eukaryotic cells, but your factory setups are definitely a bit different. Let's compare. Plant cells pack two major features that animal cells just don't have. Number 1, chloroplasts. These are basically magical little solar panels. Because plants can't exactly walk to a drive-through, they use a green pigment called chlorophyll inside these chloroplasts to catch sunlight and literally bake their own food. Animal cells don't need these because, well, we just eat our food. Number 2, plant cells have a thick, rigid outer armor called a cell wall that wraps completely around the outside of the cell membrane. Animal cells? We're squishy. We only have that flexible membrane. Which actually brings up a really cool thought experiment from the source material. Why does a plant even need that hard armor when a dog or a human doesn't? Well, think about how you stand up right now. You've got a skeleton made of solid bone. A tree or a tulip doesn't have bones. Because plants are stuck in one place and have to stand tall against gravity and wind, they completely rely on millions of these microscopic, rigid cell walls, stacking together like sturdy bricks. If a plant's cell wall suddenly became as soft and flexible as an animal cell membrane, the whole thing would just instantly wilt and flop over onto the ground like a wet noodle. Section 5, How Cells Divide. So if you start out as a tiny baby, how do you grow into a full-sized adult? Or when you wipe out on your bike and scrape your knee, how does that skin heal? The factory has to know how to copy itself. Hundreds of billions of your cells are replaced every single day through a super-controlled division process. And there are two ways this goes down. First is mitosis. This is your daily maintenance program. One cell divides into two perfectly identical copycat cells. This is exactly what heals that scraped knee and makes your bones grow longer. Meiosis, on the other hand, is completely different. It's a special two-step process strictly for sexual reproduction. It creates 4 totally distinct cells, and each one only has half the genetic information. That's what creates the amazing genetic diversity we see all around us. Imagine if your skin cells used meiosis to heal that knee. Complete disaster. The cells wouldn't be identical copies, they'd only have half the instructions, and your skin would totally fail to repair itself. But wait, how do the cells know when to actually stop dividing? If mitosis just ran on a loop forever, you'd end up with a massive mountain of skin on your knee. Well, animal cells have this genius built-in sensor called contact inhibition. Essentially, when an animal cell gets a little too crowded and bumps into its neighbors, it's like, oop, no more room, and it politely just stops dividing. It is the ultimate crowd control. But sadly, when cells mutate and lose that stop instruction, they just divide uncontrollably and pile up on top of each other. And that is exactly what causes tumors and cancer. Interestingly enough, plant cells don't even use contact inhibition because their rigid cell walls lock them in place from the start, so they have a totally different growth pattern. Section 6, the unifying cell theory. For our final section, let's zoom all the way back out for the big picture. The stuff we've explored today didn't just happen overnight, you know. It took some brilliant minds collaborating across decades. In 1838, a botanist named Matthias Schleiden figured out every single plant was made of cells. The very next year, a zoologist named Theodor Schwann realized the exact same thing applied to animals. And then in 1855, Rudolf Virchow proved that new cells don't just magically pop into existence, they only ever come from older, pre-existing cells dividing. Put it all together, and their discoveries form what we call the classical cell theory. It's basically the grand, unifying rulebook for biology. Rule 1, all living organisms are made up of one or more cells. Rule 2, the cell is the basic unit of structure and function. It's the fundamental Lego brick. And Rule 3, all cells arise from pre-existing cells. This incredibly simple set of rules connects every single living thing on Earth, from a tiny bacteria in a hot spring to a giant blue whale, directly to you watching this explainer right now. Science never really stops pushing the boundaries, though, does it? Rule 3 says all cells come from pre-existing cells. But back in 2010, scientist J. Craig Venter and his team did something straight out of a sci-fi movie. They mapped the entire DNA code of a simple bacteria on a computer. Then they went into a lab, took non-living chemicals, and completely synthesized an exact physical copy of that DNA code from scratch. They're literally learning how to write the instruction manual for life on a laptop.
Which leaves us with a pretty massive, provocative question from the text to wrap up this explainer. As we get better at building the code of a cell from non-living chemicals, what is the future of synthetic cells? I mean, could we custom build cells to eat up pollution or cure genetic diseases? And if we're creating life from scratch in a test tube, what kind of ethical guardrails do we need to set up as a society? It's a heavy conversation, and it totally proves that the microscopic world of the cell is still one of the most exciting frontiers out there. Thanks so much for hanging out with me on this journey to the center of the cell. Keep questioning things, keep exploring, and I'll catch you next time.